What are viruses? Today we can define them, describe them, but for centuries they were invisible mysteries. Curious people, driven by questions and armed with scientific mindsets, slowly uncovered their secrets. Viruses have shaped human history, they've shaped medicine and technology. Their story is still unfolding, and to understand where we're going, we need to see where it began. This is the history of virology. Here's our timeline of virology. We'll explore key figures in the history of virology from the late 1700s to the mid-1900s. In 1796, Edward Jenner observed that milkmaids who caught the mild disease cowpox didn't get smallpox, a far more serious illness. At the time, doctors practiced variolation, deliberately infecting patients with smallpox to induce immunity. To reduce the risk of standard variolation, Jenner inoculated an 8-year-old boy with cowpox first, then later exposed him to smallpox. The boy remained healthy. This was the first successful vaccination using a related but safer virus. Over the next century, vaccination spread worldwide, but the cause of diseases like smallpox remained unknown. Without the concept of viruses or the tools to detect them, progress was limited. Enter Louis Pasteur. In the late 1800s, he advanced microbiology through work on fermentation, sterilization, and disease. He developed vaccines for rabies, a viral disease, and anthrax. He proposed that some infectious agents might be smaller than bacteria, an early hint at viruses. In 1892, Russian botanist Dmitry Ivanovsky was studying tobacco mosaic disease, which was damaging tobacco crops vital to trade and agriculture. He passed sap from infected plants through porcelain filters with pores too small for bacteria to pass through, but the filtered liquid still caused disease. He suggested it might be a toxin or a very small bacterium. 6 years later, in 1898, Dutch microbiologist Martinus Beijerinck repeated Ivanovsky's experiment and showed that the agent could pass through filters, remain infectious after dilution, but could not grow in nutrient broth. He concluded it could only reproduce in living cells and called it a contagious living fluid, what we now call a virus. In 1915, British bacteriologist Frederick Twort observed something in bacterial cultures that destroyed bacteria, but he wasn't sure was it a virus, an enzyme, or something else. 2 years later, in 1917, French-Canadian microbiologist Félix Darel independently made the same observation and concluded it was a virus that infects bacteria. He named it a bacteriophage, meaning bacteria eater, and showed it could reproduce and clear bacterial cultures. In the 1920s and 30s, he used phages therapeutically to treat bacterial infections. His early results were promising, but inconsistent, due to the limited understanding of phages and lack of modern controls. Phage therapy faded with the advent of antibiotics, but as resistance builds, his work serves as the foundation for renewed interest today. In 1935, Wendell Stanley crystallized the tobacco mosaic virus. He showed that viruses could be isolated as physical particles composed of protein and RNA, confirming that viruses were not cellular organisms. This helped define viruses as biological agents distinct from both cells and chemical toxins. In 1952, Alfred Hershey and Martha Chase used bacteriophages labeled with radioactive markers, sulfur for protein, phosphorus for DNA. They showed that only DNA entered bacterial cells and directed viral replication, proving DNA carries genetic information. Their experiment was elegant and transformative, reshaping molecular biology. Beginning in the mid-20th century, electron microscopy allowed scientists to directly visualize viruses for the first time, revealing structures like capsids and envelopes. As molecular biology advanced in the following decades, techniques like PCR, genome sequencing, and reverse genetics made it possible to study viral replication, mutation, and interaction with host cells, turning virology into a precise, gene-level science. Over 150 years, virology evolved from scattered observations to a molecular science. What began with disease prevention became the study of biological particles, their structure, and their genetic code. Today, viruses are used in vaccines, cancer treatment, and gene therapy. Does any of this change how you think about viruses? This has been the History of Virology.